Hello everyone, let us discuss about the various genetic terms that you should know in the genetic biochemistry. So before starting the various genetical terms you should know about the biochemistry, in genetic biochemistry, I want you to know about the concept of alleles and concept of the loci. So if you don't know the concept of allele and loci, I want you to read my previous lecture. And before going towards the genetical terms, I want to describe you about the concept of the genotype and concept of the phenotype. Concept of the genotypes and concept of the phenotype. What do we mean by the genotype and phenotype? The genotype is the genetic print of the human body. So the phenotype is the external appearance of the human. So what are the imprint that is present in the genes? That is known as the genotype. And what are the various physical characteristics that we see from our eyes? That is known as the phenotype. So knowing this concept, I want to start the various genetic terms. So first is codominance. The codominance, in the codominance, the both alleles contribute to the phenotype of the heterozygote. For example, the blood group A, B, AB, the alpha antitrypsin deficiency, and USMLE groups, the human leukocyte antigen groups. The another is variable expressivity. Variable expressivity means the patient with the same genotype, having multiple phenotypes. That means having varying phenotypes. The patient may have the same genotype, but he could have the different phenotypes. For example, the patient A, there is one patient A and patient B. The patient A is also suffering from neurofibromatosis type 1. The patient B also may be suffering from the neurofibromatosis type 1, but they may have varying disease severity. The patient may have no skin lesions at all, but the patient could have the multiple skin lesions, the multiple neurofibromatosis lesion in his skin. So this is one of the examples. Another is incomplete penetrance. Not all individuals with the mutant genotype show the mutant phenotype. The percent of penetrate, which is multiplied by the probability of inheriting genotype, is equals to risk of expressing the phenotype. For example, the BRCA1 gene mutation doesn't always result in the breast or ovarian cancer. One may tell that we always say that I smoke a lot of cigarettes, but I don't have the carcinoma, I don't have the lung cancer. I have my mother died of the breast cancer or ovarian cancer, but I don't have breast cancer or ovarian cancer. It is the miracle of God, but it is not that always, because it is of the incomplete penetrance. If the person could have the complete penetrance, then there could be the complete expression of the phenotype, because not always mutant genotypes results in the mutant phenotype. So if you have the mutation in your gene also, but you could not have, but there is also no chance of expressing the phenotype. For example, if you have the BRCA1 gene mutation, but you may not contract the breast or ovarian cancer in your lifetime. This is the concept in the incomplete penetrance. So let us move towards the Pleiotropy. The Pleiotropy, one gene mutations contribute to the multiple phenotypic effect. For example, untreated phenylketonuria. If the phenylketonuria is untreated, then the person could manifest into light skin, intellectual disability, and musty body odor. So these are the multiple phenotypic effect of one gene mutation. So this is the concept of the Pleiotropy. So now let's move towards the other genetical terms. Let us discuss now anticipation. So what is anticipation? Anticipation is the increased severity or earlier onset of the disease in the succeeding generation. For example, Trinucleotide repeat diseases. One of the diseases is Huntington disease. The Huntington disease may be the less severe in the first generation, and the onset of the disease could be very low in the first generation. For example, the Huntington disease may not be very severe in the first generation, and the onset of disease could be, for example, in the late 40s or late 50s. Just I am assuming. And but in the second generation, there could be the appearance of the few symptoms, and the disease onset could be of early 20s or early 30s. And in the third generation, there could be the severe appearance of the symptom, and in the third generation the onset of symptom could be in the early 10s or early 20s. So the disease severity is also high, and the onset of disease also becomes earlier. That means it is known as the anticipation. So loss of heterozygosity. What is meant by the loss of heterozygosity? If a patient inherits or develops a mutation in a tumor suppression gene, the complementary allele must be deleted or mutated before cancer can occur. But this is not true in the case of oncogenes. For example, retinoblastoma and the 2-hit hypothesis, and in the Lynch syndrome, in the Li-Fraumeni syndrome. It is known as the loss of heterozygosity. And dominant negative mutation. What is dominant negative mutation? If there is dominant negative mutation, there exerts a dominant effect. For example, a heterozygote produces a non-functional altered protein that also prevent the normal gene products from the functioning. For example, we know that p53 is the tumor suppression gene, and when there is the single mutation in the p53 tumor suppression gene, then there is the production of a certain protein. That protein binds to the DNA and block the normal functioning of the another normal p53. The normal functioning of the p53 is that it binds the promoter to prevent the further increase of the carcinomas. We know the initiator and the promoter are the key role in the occurrence of the carcinomas or any other cancer. So the p53 binds to the promoter and it renders it inactive. But if another p53 tumor suppression gene is mutated, then it also affects the normal functioning p53, which hinders in the normal gene function. So this is the concept of the dominant negative mutation. So now let us move toward the linkage disequilibrium. So now let us discuss about the linkage disequilibrium. Linkage disequilibrium is the tendency for certain allele at two linked loci to occur together more or less often than expected by a chance. So it is measured in the population, not in the family, and it often varies in a different population. So this is the concept of the linkage disequilibrium. Now let us go towards the mosaicism. The presence of genetically distinct cell lines in the same individual is known as mosaicism. So there may be two types of mosaicism. One is somatic, another is gonoidal. The somatic mosaicism. Let's talk about the somatic mosaicism. The mutation arises from the mitotic error after fertilization and then propagate throughout the multiple tissue or organs. It is known as the somatic mosaicism. It means there is the direct visualization of the mutation in the physical form. Whereas in the gonadal mosaicism, the mutation only occurs in the eggs or sperm cell. If the parents and the relatives do not have the diseases, then we suspect then he or she have the gonadal or germline mosaicism. So the parents doesn't have the disease and it is not manifested in the physical form, then he or she could have the germline mosaicism. Now let us discuss about the McCune Albright syndrome. The McCune Albright syndrome occurs in the GS protein activating mutation. If there is the GS protein activation mutation, then there is the McCune Albright syndrome. It is present with the unilateral cafe au lait spots. I will show you the picture in the subsequent lectures. So what happened? There is presence of unilateral cafe au lait spots with unilateral cafe au lait spot with ragged edges and polystotic fibrous dysplasia. It means the bone is replaced by the collagen and fibrous tissue. So there is at least one endocrinopathy. So at least one endocrinopathy. For example, precocious puberty. It is lethal if the mutation occurred before the fertilization, but survival in patient with mosaicism. So if it is present in the gonadal mosaicism, then there could be the chance of survival because it is only present in the germline. Germline mutation only occurs, it doesn't occur in the physical form. So now let us discuss towards the locus heterogeneity, allelic heterogeneity, heteroplasmy and uniparental diasome. Now let us discuss about the locus heterogeneity. The locus heterogeneity means the mutation at different loci can produce similar phenotype. For example, albinism. So here is the gene. So in the one loci there is the mutation, and if mutation occurs in another loci and there is the production of the similar phenotype, then it is known as the locus heterogeneity. And what is meant by allelic heterogeneity? Allelic heterogeneity means different mutations in same locus can produce same phenotype. For example, beta thalassemia. For example, it is the gene and mutation 1 occur here and mutation 2 occur in the same locus, and there is production of the similar phenotypic features, then it is known as the allelic heterogeneity. And what is known as heteroplasmy? The presence of both normal and mutated mitochondrial DNA resulting in the variable expression in mitochondrially inherited disease, because all mitochondrial DNA is passed from mother to all the children. So it is known as the heteroplasmy. What is the uniparental diasome? The uniparental diasome. We know that normally what happens that one copy of chromosome comes from the mother and one copy of chromosome comes from the father. So both copies of chromosome come from both the parent. But uniparental diasome means two copies of chromosome come from the one parent. And heterodiasomy means meiosis 1 error and isodiasomy means meiosis 2 error. The uniparental is euploid. The most occurrence of uniparental diasomy results in the normal phenotype. So consider isodiasomy in an individual manifesting a recessive disorder when only one parent is the carrier. For example, Prader-Willi and Angelman syndrome. So it is one of the examples of the uniparental diasomy. So these are the most important genetic terms that you should know in the genetic biochemistry. Thank you.